So children, we continue with what we had done in the previous class, that is DNA, the structure of DNA I had done, that's it. So I'll continue with that. I'm not staying in DNA anymore. I have to do cell DNA replication, but I'll be doing while teaching the cell cycle. Okay, so what term did I use? Cell cycle. Okay. So in this particular chapter, we have to do two types of cell division, that is mitosis and meiosis. We have to do mitosis and meiosis. Right now we are doing with mitosis, though meiosis also the phases what I'll be explaining, G1s, G2 phases occur, but it doesn't enter into cell cycle again. So just now let us concentrate what happens in mitosis strictly. Is it clear? So mitosis is a type of cell division which occurs in the somatic cells. So first let us do with the cell cycle, then I'll come to the detail of mitosis. So what is a cell cycle? Let me first explain. Cell cycle, if we draw cell cycle, we can show it like this. So here the cell cycle, since I am doing mitosis, it is divided into, so what are the phases it is divided into? The cell cycle. This is a division phase. What is the meaning of division? This is the phase when the cell is dividing, division phase. We also call it the mitotic phase. We can also call it the mitotic. What is mitotic? Mitosis is the cell division, mitotic phase. This is followed by the longest phase of cell cycle and we call it the G1 phase. G1 phase. Is it all right? So here this is the part where I have highlighted, this is the part where the cell is dividing. Is it all right? Then G1 phase. G1 phase is followed by S phase and then G2 phase. Is it all right? So what does G stand for? G stands for growth. And what does S stand for? S stands for synthesis. So growth 1 phase. The first one is growth 1 phase. Cycle, since I am talking about cycle, then why we are taking G1 as the first phase? I'll come to it. Growth 1 phase. It is followed by S phase. After S phase is completed, it is a short period of G2 phase, and after the G2 phase is completed, the cell is now ready, big enough, ready, mature to divide. The cell is ready to divide. Is it clear? It has grown, it is matured, so that it can divide. Now after this G2 phase comes the division phase. Is it clear? And here the cell divides. During this phase the cell divides. All right. This phase I'll do later. This phase is again divided into two parts. In one part the nucleus divides, nuclear division, and the other is the cytoplasmic division. Cytoplasmic division, this we will do later. So here at the end of, so what is happening? The cell is growing. In the synthesis part, in the S phase, it synthesizes DNA, it replicates DNA, again it grows, now it is ready to divide. The cell at the end of division phase, what happens? Two cells are formed. After the division, two cells are formed. Is it clear? Two identical cells are formed. We call them as daughter cell, and each of the cell individually undergoes this cycle again. Individually, this will undergo G1S, G2 phase. Is it clear? This will again carry out G1S, G2 phases. Is it all right? And then each will individually divide to form new cells. Is it clear? Now what happens here? In this, why are we keeping G1 phase as the first phase? Because the new cell which is formed, it will start with G1 phase. Is it clear? And then S, then G2, and then division. The new phase, usually what terms do we use? This cell which is about to divide. Is it all right? We usually call it as a mother cell or a parent cell. Why do we use a female gender? Because females capacity of reproduction is basically for the females. Is it clear? There is no as such cells have no sex as male and female. Since it has a capacity to divide, we call it a female, means we keep it as a mother cell, and the two cells which are formed, they are the daughter cells, because they will again further divide. Is it all right? There is no mother, like no gender for it. So what happens? So we start with G1 phase. What happens with it? Now a cell, G1 phase, is it? And we will mainly concentrate on the nucleus. In G1 phase, we see the cell. This is a newly formed cell with a prominent nucleus, with nucleolus and all. We are not like bothered about it. There is a pair of centriole. All right, we will see it later, with asters around. You have done in class 9 all these things. Is it clear? And what do you see inside it? You will find there is a network of thread-like structure. And what do we call it? We call these as the chromatin fibers. We call this as chromatin fibers. Is it clear? Okay. Now, how is chromatin fiber? If in the previous class I have told you, if you don't remember, please refer to the previous previous class video. What is chromatin fiber? Chromatin fiber is coiled DNA, if you remember. Is it all right? Chromatin fiber is coiled DNA. We are not going to chromosomes yet. And I told you in the previous class that throughout this phase, such a one term I am just missing out, throughout the non-dividing phase, when the cell is not dividing, the chromatin fibers, the DNA remains as chromatin fibers. Chromosomes are not formed. Is it clear? Achha, what term do we give this non-dividing phase as? We call this as the interphase. When the cell is not dividing, what term do we give? Interphase. So G1 is a part of interphase, S is a part of interphase, G2 is a part of interphase. Is it all right? Now so we are basically doing with G1 phase. Here the chromatin fibers, these are no chromosomes are visible. Each chromatin fiber is made up of one strand, one duplicated strand, one strand of DNA. Is it clear? One strand means one DNA strand. How does the strand look like? Like this. Each chromatin fiber, is it all right? Of course, it is coiled around the histone protein. Since I am using chromatin fiber term, that means it has already coiled around histone proteins. It has turned around histone proteins. Is it clear? Chromatin fiber term I am using. Is it all right? So here are each of the chromatin fiber is made up of one DNA molecule. DNA is a macromolecule, is a large molecule. Why? Because it is made up of many nucleotides. You know what are nucleotides? Is it clear? It's a large molecule. So now let us see, you must be knowing that how many chromosomes do we have? How many chromosomes do we have in our cell? We have 46 chromosomes. Is it clear? And what are chromosomes? They are coiled chromatin fibers. That means the number. And what are chromatin fibers? They are coiled DNA. Okay, so DNA, number of DNA is equal to the number of chromatin fiber is equal to the number of chromosomes. So if we have 46 chromosomes, we have 46 chromosomes. If we have 46 chromosomes, that means we have 46 chromatin fibers as well. Chromatin fibers as well. That means we have 46 DNA strands. Is it all right? So why I am saying this, because I just want to tell you that each of the chromatin fiber is made up of one DNA. Is it okay? So here what are we finding? It is, and you cannot, it appears as a intermingled mass of thread, and we call them as chromatin fiber. Each of the chromatin fiber is a strand of DNA. You will keep in mind, because we have to show in S phase, I have to show you DNA replication. Okay, now here why it is called growth. Now during this phase, the cytoplasmic content increases, the cell grows in size. That is of course not here we have to actually concentrate on the nuclear changes, but since we are doing it, so the cell grows in size, the cytoplasmic content increases. The cytoplasmic content increases. It synthesizes lot of protein. It has to make lot of enzymes. Enzymes are made up of protein. It synthesizes lot of protein. Why are these protein and enzymes needed? Because here one important event has to occur after G1 phase. An important event has to occur in S phase, that is DNAs. How many DNAs I said? 46 DNAs. All 46 DNAs will replicate. Is it clear? And replication needs lot of enzymes. So those enzymes are synthesized during G1 phase only, because it is preparing G1 phase, the cell is preparing to undergo S phase. S phase, why it is important? Because DNA will replicate, centriole will replicate. Is it clear? So let us now come to this part, the S phase.